Hello friends, welcome to another video tutorial from Shomu's Biology, and in this video tutorial we'll be talking about plant anatomy and plant physiology. Now uh, in my channel I don't have much videos about plant biology, so this is going to add some more videos regarding it. Actually, I'm not using my own PowerPoint. I'm using the PowerPoint prepared by Edgar Moctezuma. Hope I pronounce his name correctly. And I found this PowerPoint, and it's very important, very good. Actually, I combined some other slides and some other pictures and stuff, but most of it was taken from the actual content developer. The name is provided, so it's really privilege for me to use this PowerPoint and explain the stuff because it's beautifully presented. So let's begin with it. Now, before going into the details, I must tell you about what is anatomy and what is physiology. You need to know about this stuff. That is, what is anatomy and physiology. Anatomy, let's begin with that. Anatomy means there is something to deal with the structural features of anything, like in case of plants or animals. Anatomy deals with the structure of and the morphological structures of that organism, while physiology deals with the functionality of those structures. These are the terminal differences. Now, in plant anatomy, we'll be talking about cells that make up plants, different types of cells that are present there for plants, different tissues that those cells ultimately produce. Because if you look at the hierarchy of an organism, it will begin with say cells, and if you combine some cells, it will give you tissue. If you combine some tissue, it will give you some organs. And now, if you combine organs, it will produce an organ system, and finally the organ systems together give you the organism itself, like a plant or an animal, whatever it is. So these are the stuff belonging to the anatomy part for a plant. In the physiology part of a plant, we'll be talking about the water and sugar transport inside the plant, as well as we'll be talking about the plant hormones, different types of hormones that are important in the plant body, and what are their different functionalities and all the stuff. So let's begin with it. First things first, there are something astonishing about plants that always keep us involving in studying plant biology very well. And some of them is the plant variety, and it starts with a very small variety of plants, like as you see in the left hand side in this image. It's very small variety of plants, as you can see. 1 minute. Let me take the color here. Okay. So here you can see that very small plants, like algae as are always the plants, and also you can see this large trees in the rainforests. So all of these are plants. So much versatility in this complete plant kingdom. So much different versatilities we can see. So let's see. Again, what is plant anatomy? It is the study of structure of organisms, looking the cell and tissue structures, how they are constructed and how they are arranged. The study form is known as the plant anatomy. Well, plant physiology means the functions of all those cells, tissues and organs of the living organisms. So I've already talked about it. Now the thing is, structure correlates with the function of the things. In real life, in biological science, it's always true. Structure correlates with the functionality, because if you look at the core of every organism, you will find it's built with proteins, ultimately built with some kind of proteins. Now, if you take those proteins, you will see the structure of the proteins influence the functionality of the proteins in huge impact. So the thought of the day would be, how could be this large trees, this very tall trees, get water from the ground till the top? This is one of my questions I used to ask in my childhood time. That how could these big trees get water from from the soil till up to that top of 100 meter, even more than 100 meters, 200, 250, 300, even more than 300 feet height. So example, the redwoods usually take. So we are going to study all these physiological processes of how plants take up water from the soil, as well as how the food it produces in the leaf slowly start to spread throughout the plant body for the cells to live on. So how those things actually is possible, we are going to talk about that. Now, if you begin with the anatomy of the plant cell, it is the first thing that in the plant anatomy we'll be talking about first about the cells that are making plants. What are the cells that we know of? Here, plant cells are basic building blocks like any other animals, any other organism cells, because cells are the building block of organisms, and they're specialized and they have a specific function. There are specific type of cells present for specific type of functions in different plant body. Okay, as you see, there are three different types of cells are provided here. Some cells are filled with only vacuoles, nothing else to do with their functionalities, to have the vacuoles and store some of the stuff in the vacuoles and stuff. Some cells contains a lot of chlorophylls like this one, which will produce food after photosynthesis, and some of the cells, as you see, they are floating in the water, so have different natures of the cells. So you see it, by working together forming tissues, they can support each other and they can survive, and as the cells survive, the organism is also surviving. So if you look at the level of organization, it begins with atoms. Atoms form molecules, molecules form cells, cells connect together to produce tissues, multiple tissues together produces organs, and organs will combine together to give us a whole plant, and that's how it works. Now, if you look at different types of plant cells, you will see it here. There are three major types of plant cells that we are going to talk about. One is the parenchymal cell, second one is collenchyma, and third one is sclerenchyma. So parenchyma is the first type, and it is called the normal plant cells that have large vacuoles that we already saw in the earlier video, that large vacuoles, no secondary cell wall, and usually not mitotic in nature, but they can stimulate and they can divide when they are stimulated by any sort of injury in the plant. So if you injure a plant, this kind of parenchymal cells may divide after that stress response. There's the signaling pathway inside the plants going on while the stress response is occurring, and that stress response will help parenchymal cells to divide and act as a stimuli to divide. And what are the examples? Example means here you can see most of the cells that make up the ground tissue of the plant cell called the parenchymal cells. Now, what are the ground tissue? We haven't talked about it yet. We'll be talking about it slides later, some 4 or 5 slides later. So you'll know what is it. But for now, ground tissues are mostly made up with parenchymal cells. And parenchymal cells, there can be photosynthetic cells in leaf mesophyll. There can be cells that store starch and stems in the roots. As you know, potato is a type of such kind of cells that can store starch. Fruit cells can also be like store sugar, or sieve tube cells that make up phloem. So parenchymal cells are very versatile. They can make phloem cells, they can make cells to carry photosynthesis, they can make cells to store starch. Second one is the collenchymal cells. You see here the difference between parenchymal cell and collenchymal cells are given in this picture. You see these are the cortical parenchymal cells and these are the collenchymal cells. Parenchymal cells are larger, you can easily view it, kind of transparent in nature, filled with vacuoles, while the collenchymal cells are constricted. Their cell wall is very, very thick. And you see collenchymal cells they are elongated, thicker primary wall but no secondary wall at all, and it provides the growth and the support of that organism. Examples: cells that support young stems and petioles, and some ground tissue can also be made with collenchymal cells. Third one is the sclerenchyma, completely different type. These are rigid secondary cell walls can be found there. So parenchymal cell as well as collenchymal cells, both of them do not contain any secondary cell wall, but sclerenchymal cells contain secondary cell wall. Not just they contain, but they contain very rigid and thick secondary cell wall, and they may be dead and used for plant support. So that is the major functionality of sclerenchymal cells, to provide a very important plant support. Now, examples for sclerenchymal cells are the shells of nuts and outer coat of the seeds, and fiber cells that used to make plant fibers that are used to make rope. Those are the cells that make the sclerenchyma. Sclerenchymal cells are they're kind of fibrous in nature, elongated in nature, and they're very, very strong in holding tension and strength. So we use sclerenchymal cells a lot for our purpose to produce rope and stuff. Now here are some images of all those type of cells. These images already been shown. Now, these are the examples of collenchymal cells and parenchyma. You see the parenchyma, it's always large, big, easy to see, filled with vacuoles, while collenchyma is having a thick wall between those cells. And as you see, these are the sclerenchymal cells. Sclerenchymal cells are elongated, they have secondary walls, a lot of things surrounding the cell cytoplasm actually. So thick secondary wall. This complete section, these are the sections of secondary cell wall. Okay, so you see that. Now let's focus on plant tissues. We know what are the cells. Now it's time for knowing the tissues. Now plant tissues, it begins with three different types of plant organs that we want to talk about: roots, stems, and leaves. Now in this lecture, we'll divide the lecture in this way that we will start talking about roots, stems, and leaves. So you begin with the different types of tissues that are found in both on all these type of organs. It begins with first type of tissue, it's called the dermal tissue. They are the outermost layer of a plant cell. And second type of tissues are the vascular tissues. Vascular tissues are conducting tissues, and they are helping in transport of water as well as transport of sugar. They are called the vascular tissues. Example, xylem and phloem. You probably heard those names a million times before. Third one is the ground tissues, which are bulk of inner layers that is present, and most of the cases ground tissues provide either stock solutions like stockage of starch and materials for the future use, or the ground tissues may help in the strength and proper support for the plant. Now begin with the dermal tissues. You see here, dermal tissues are always the outermost layer of the plant cells. You see the dermal tissues will make this root hairs and those things, cuticles that are coming out completely surrounding of the cell, and that is very, very important because dermal tissues kind of prevent plant for different infections as well as many wound and all the stuff, because it acts as a skin of plant like us also. The dermis, example epidermis, it's the outermost layer of the cells, and you see like the skin of animals, I talked about it. And in stems and leaves, there is this very thin waxy layer that is found coating plant leaves, and those coatings are known as cuticles, and the cuticles are the prime example of epidermis, such as dermal tissues. Some have trichomes or hairs. You see some of the plant leaves may have trichomes or small hair protrusions coming out, or sometimes the roots also have, not sometimes, root definitely have this kind of protrusions coming out. They are called root hairs. Now second type of tissues that we want to talk about are the vascular tissues. And vascular tissues, the matter you think of vascular tissues, you are thinking of the tissue that are helping plants for taking water from the soil till the top, as well as transferring the sugars that are produced in the leaf throughout the body of the plant cells. These are the vascular tissues. Example, xylem and phloem, both the way they work. Xylem transport water and minerals and dissolved ions from the root to the stem and the leaves. So it takes something from the soil till up. So take from the root till up towards the leaves. While phloem carries dissolved sugar in the water from the leaves to rest of the plants, so downwards. Okay. Now let's talk a little bit more about xylem and phloem, because they are very, very well constructed tissues and they're worth talking them separately. So xylem transports water and dissolved minerals from soil through the root up to the top of the plant, and they have tracheids. Tracheids, different cells are present in xylem to make xylem tissue what it is. And similarly, phloem contains different types of cells that make phloem. Now, xylem contains a tissue called tracheids. Tracheids are long, thin, tube-like structures without any perforations at the end. So you see here tracheids, the image is not provided, but tracheids are very thin structures, as you know, kind of fibrous in nature. So it's kind of fibrous in nature. It is a very thin tube-like structures without any perforations at all these ends. So it's kind of fixed like a tube, and those tubes are arranged one after another to give the structures of a xylem properly, because you need a tubing like a pipe to carry water from the soil till top of the plant. Second one is the vessel elements. Vessel elements are shorter versions, and these are short, wide tubes perforated at the ends. So it will look something like this. It will smaller tubes, and they have perforations at the end. Both the end contains perforations through which the water and minerals, dissolved water, can pass. Okay, and both of them, tracheids and vessel elements, combine together form what is called a pipe, right? And they call it the vessel pipe. Through the vessel it can go, because you know, in the formation of pipe is important, and it needs to have a specific pore in the middle so that water can take and water can pass, and obviously it needs to have some solid state of structure to give its structural integrity, and both of these things played by tracheids and vessel elements together. That's why they patch up very well to give xylem a proper structure. Now both the cells have pits on the walls. Okay, pit means thin sections that are present in their walls. So these are the examples. I draw some of them. As you see, this is the example called tracheids, and these are the vessel elements and tracheids, and both of them contain pits, smaller thin sections in the wall that is always present to interact between the nearby cells. Okay, now xylem cells are actually dead. You probably astonished about that. Why xylem cells are dead? Actually, you thought it's going to be alive because it's carrying water. No, it's dead because it's not required to divide and multiply xylem cells. Why would you require? Because it's not a physiological unit. It is a kind of very steady physical function, and you don't require the cell to grow and divide for this functionality. Now it was active and alive few years back. That's why the cell is elongating as the cell is evolving and the plant is growing tall and tall. At that time cell is dividing and these things are evolving, but after some time those cells become dead, and those dead cells accumulate to form xylem through which water and minerals can pass. Right, they're just nothing but the hollow cells and consist of only cell wall, nothing else. As you see here, this is the early formed xylem which were active in nature. As you see, as the plant is growing, the top portions, the shoot, if you look at the shoot structure here, you see these are the shoot structures coming out, the leaves and all the stuff. Okay, so this is kind of looking like this. So xylem, as it is forming, as the tree is moving and evolving, and it is growing in the top, at that time, you know, the xylem cells become active and actively dividing to create the large pipe, because you need to construct the pipe as the tree is going on, as the tree is growing top, getting the height. But after some time, all those cells are dead and becomes a hollow chamber, it's a hollow pipe without a very thin cell wall. Second type of this vascular tissue is phloem, and phloem is also very important because it has the function to carry dissolved sugar from the plant leaves through all the cells of the plant body. So phloem cells transport the sugar, and phloem cells are alive, unlike the xylem cells. And if they are alive, and in this case they lack nucleus and other organelles, because those things are not found, because they don't require to function there. These cells are kept there for the functionality of transferring the sugar to all other cells. They are another type of branching pipe elements that is present in the tree to deliver the sugar and the food to all the cells for the cells to live. You see here, as you see, these are the structure of a phloem, and you see there are companion cells found in the phloem. These are the phloems. Long, large phloem tubings are present. It's kind of seeing like it's dead, but it's not dead, it's actually alive. And they don't have nucleus and more often organelles, but they have sieve plates. Sieve plates is like a sieve, just to figure out and sieve out all those things that are required to pass, and some things will not be passed, so they will be blocked using the sieve plates. So sieve plates are required for specific things to block to be flown, and specific things to allowed to be flown to the next place. So here these are the phloem parenchymal cells, large cells they are covering, and also they have companion cells surrounding, and those companion cells are alive, they contain nucleus. That's why phloem cell requires companion cells, because phloem cells don't have nucleus, don't have other organelles. So for any other requirements, for any other functionalities, they need to rely upon some other cells, and there is the functionality of these companion cells for the function. So phloem transports sugars and the phloem composed of cells called sieve tube members, or it's known as STM, sieve tube members, because they have sieve plates between two different tubes. If I draw it, it will look something like this. It's kind of sieve plates, and then another tube starts, right? And companion cell joint sieve tube members are related and help to load materials into the sieve tube members. So the companion cells will stay there. They will do all the functionality of getting because they are active. Their functionality is completely very fast, like a live normal cell. So companion cell will take that sugar and it will transport and put the sugar to this sieve tube members, or STM, put the sugars to the phloem, so that phloem can bring that sugar from the plant leaf to the rest of the body, rest of the regions. The end of walls of STM have large pores called sieve plates. So we have talked about sieve plates earlier. We've seen the images of sieve plates. Sieve plates are present at the end of each of the phloem tubings. So get the idea of this phloem. The phloems are nothing but a pipe or a chamber, as you know. This one, a hollow pipe to carry the sugar, the dissolved sugar, which every cell requires to survive in the plant body. And in this case, this companion cells are finding the process very easy. Companion cells doing the process, they transfer the sugar with water to those tubes, and the tubes are flowing it only, and they have sieves in the middle to filter it out properly. So these are sieve tube members and these are companion cells, and we know that, and the sieve plates. Third kind of tissues, finally, they are called ground tissues. Remember, ground tissues are found, they are bulk of the plant. They make most of a plant body, right? And whatever thing you know of, the wood that you know of, the hard thing, actually made up with ground tissue. It's not xylem, it's not phloem, or it's not any other of this endodermal layers, exodermal layers, or epidermal layers, sorry. They are all bulk ground tissues. And in this case, their functions can be metabolism, storage, or support. Mostly storage and support. The example for storage is in a potato. It's filled with starch. And the support, it also provides supports for redwood tree, if you take an example. So see, if I dissect the stem and root, both of them together, you'll see the ground tissues very well. And this is leaf. All of them contain that tissue. So ground tissues are found everywhere. For every single different regions of the plant anatomy, we need to have a ground tissue to provide some support, as well as to provide some storage element if it's required. Mostly it's found in the roots as well as stems. Redwood example, stem is a ground tissue containing element. In roots of potato is an example. In leaves, there are some examples I don't remember right now. So these are the examples, as you know. If we break them down in roots and stems and leaves, you'll see leaf contains dermal vascular ground, roots also contain that, as well as the stem contains that. So every single section of plant body, the anatomy, contains all the three types or three varieties of tissues, and in those tissues composed of different types of cells belonging to parenchyma, collenchyma, and sclerenchyma. Okay. So functions of the plant organs. So if you begin with this different plant organs, roots, stems, and leaves, because we divide them in three different parts to talk about them in different manner in much more details, because I've told you I'll divide this in a way to understand more easily. Roots are the anchorage unit which helps in associating and taking up water and minerals from the soil, water absorption from the soil, storage, and water and nutrient transport from the soil till every cell of the plant body. While stems give the support and water nutrient transport also, while leaves do the photosynthesis, production of foods. So let's move on and check their functionalities. First is root. So what are roots and what are their functionalities and their structural features? Roots are the hidden half of the plant body. Now, if you ask very little children about the plant body and plant, they will also talk about the top part that we can see from outside. But actually, we see half of the plant's anatomy. Another half, which is very important, that is roots. that we didn't talk about very well. So here root is the other half, and the function of the roots involved in the anchorage of the plant. It involves the absorption of water and minerals from the soil and transport it to other cells of the body. It can also act as storage solutions, storage for sugars and starch. It can have a conduction of water and nutrients. And if you look at the anatomy now, you'll find beautiful images, and actually these are some real sections of the plant roots. If you take plants, if you take a sibling, and if you take a root cross section of the root, you'll see these images. And if you have to stain them with different stains to see them in multiple colors. Now here you see the anatomy of roots can be varied from different types of plants, because in this case this plant contains a very congested kind of structure, as you see, but in this case it's kind of very basic structure. At the very middle, you see this is the structure constructed. This is, again, if I zoom in, you look structure like that. Again, in another plant, if you zoom into this plant, you'll see a structure like this. Now the structure at the very middle, very core of those plant roots are made with vascular cells. They are called vascular cylinder because they're made with vascular tissues of the plant. That means they contain vascular cells like phloem and xylem. So xylem and phloem produces the vascular cylinder in plant roots. That is found in the middle, and it's very, very well recognizable compared with the rest of the region of the root. And as you know, this is the vascular cylinder. The outermost layer is known as the epidermis. This is the dermal layer, right? The derm tissue that is present outside. And the third thing is the cortex that is present in between the epidermis and the vascular cylinder. It's called the cortex, and cortex is made with multiple tissues. And the cortex actually contains several different, mostly ground tissues to give the wood structure and storage unit most of the time. So this is a schematic representation of a plant root, an epidermis, cortex, and in the middle vascular tissue. You should not forget this basic diagram. And if we talk about root epidermis is a very important thing, because from this root epidermis their protrusions start to come out, and they are termed as root hair. And root hair plays a vital role in increasing the surface area of a root so that it can uptake more water, it can absorb more water and minerals from outside. And also it protects the root from diseases, because soil is a huge reservoir of microorganisms, and it can easily infect the plant tissue if there is no outer skin-like layer like root epidermis. Now, we talked about that. The root hairs are nothing but those epidermal cells and simply the extension of those cells. So simply, if you look at here, this is the normal root shape and this is the epidermal layer. You'll see an extension of that epidermal layer, extension of the cytosol of that epidermal layer. That cytosolic extension is known as root hair. Now, they increase the surface area, and it helps for better absorption of nutrients and water. Okay, so if you look at here the real image of a cell cross section, it's not a cross section, it's longitudinal or side image of the cross section. You'll see this is the cell, and this is just a cytoplasmic extension of those cells that is making the surface area more, because for these small root hairs coming out, you'll see a lot of surface area surrounding those root hairs. And water mineral absorption becomes much more easy with root hairs. And not only that, root hairs also help for some kind of mutual integration of bacteria with plants. We see these things in case of agrobacterium. Agrobacterium is a type of bacteria which tends to interact with those kind of plant root hair, and they will enter through root hair and cause an infection to the plant. It's like a tumor formation of crown gall disease. Now, crown gall disease added two benefits, kind of mutual understanding between the bacteria and the plants, because bacteria gets the nitrogen source from the plants, while the plants are getting some important factors from this bacterial cells. Okay. Now, if we talk about root cortex, that is the middle portion of the epidermis, and so cortex means, this is the epidermis, this is the vascular layer. So rest of the things are known as root cortex. So if you see, root cortex are sources of starch, sugars, and any other substances that we know of that are delicious to eat. Like as you see, all the images are given here. They are beautiful things and we tend to love to eat them. Example is cabbage, right? So root ground tissue. The ground tissue is the tissue which actually produces that storage unit where we store all those starchy materials in the root, okay? And they provide support. They have the storage unit to store starch and other sugar forms like yams, sweet potato, and other stuff. So the cortex is surrounding the vascular cylinder, and that cortex can contain all the structures as you know of. Sweet potato is not what we want here. This is a potato which is filled with starch. Where? Now if we talk about the endodermis, there is a term called endodermis in the root cortex. So if I give you the structure, you know this is the root cortex, but where this is the epidermis layer, this is the epidermis layer, this is the cortex, and the cortex is interacting with the vascular cylinder in the middle. Now the region where cortex is interacting with the vascular cylinder region, there is a specific set of cells present, and that is known as endodermis of the root cortex. And also there are specific type of structures found called Casparian strips in the root cortex. What is Casparian strip? Casparian strip is water impermeable strip, kind of waxy material in the endodermis that we saw earlier. The endodermis region. Why these are different cells? Because the cell contains a specific type of waxy layer materials coating so that water should not enter or get out from these regions, as you see in this picture. This also helps to control the uptake of minerals into the xylem, and they have to go through the cytoplasm of the cell wall, because in neither way they can go through the cell wall. They need to go through the cytoplasm of the cell. As you see, small strips that are found kind of lining all the cells that are present. So you see, epidermis layer, this is epidermis layer, this is the cortex, and cortex is interacting with the vascular cylinder, and these are the sieve cells. These are the cells with these black points, and these are known as the Casparian strips. That's why we find a specific ring around the vascular cylinder there. If we talk about stems now, because you talked about roots, now let's talk about the other half of the plant body, because stems carry a lot of things. Whatever we see about stems, because you don't see roots, whatever going on underground, but we see things on top of the ground pretty easily. It carries the fruit, it carries the beautiful flowers, it carries the leaf which will produce the food. So here in the same, you see the above ground view, and we're talking about, in this case, it supports the leaves, support fruits, it provides the aesthetic beauty, and also it helps in the reproduction of the plant, right? Because it carries the flower. And it also has some very important functional role like conduct water and sugar and all those stuff. So if you look at the stem anatomy, and if you look at the schematic of stem anatomy, you'll find that it contains dermal tissue, ground tissue, and vascular tissue, all of them together planning to have this structure. And we have the outer layer called epidermis, just like the roots, but in this case we not always find the extensions, but there are extensions. If you see trees very carefully, not all the wooden trees but other type of trees like green trees and leafy trees, you will find small extensions coming out in some cases. Like if we talk about tomato plants, you will see small extensions coming out from this epidermis layer. On the hand, we have a very center is big, because in this case we require a lot of region for our vascular bundles. And vascular bundles are not placed in the center like the roots. Here the vascular bundles are present, they are surrounding a specific region in the circle, and they are present in huge amount because they need to carry a lot of water, they need to carry a lot of minerals, and if necessary they need to carry a lot of sugars and bring it to other cells inside that plant. So here you know, in this case, see epidermis, cortex, and here we have the vascular bundle cells. Vascular bundles consisting of xylem and phloem, and they present in high amount here. The type of stems that is present here, the type of stems that we require is a monocot stem or dicot stem are present. You know, in the roots they are also varying, like monocot root and dicot root. We didn't talk about it much, but here in the stems there are some types. In monocot stems they have a specific feature, in dicot they have a different type of feature. Like in monocot stems, the vascular bundles are found scatteredly in the center portions. Like if I draw a schematic, you'll see the epidermis, the cortical layer, and here is the scattered sections of the vascular bundle. But in the dicot, you'll see the scattered thing. In the dicots, you'll see the vascular bundles surrounding a specific circle only. This is dicot and this is monocot. Okay. So type of stems again, herbaceous and woody. We talked about herbaceous stems, and herbaceous stem, if you look at the herbs, you will see that epidermal layer contains protrusions like root hair, and these are extensions for respirations as well as for photosynthesis, but very less for photosynthesis, but more for taking up water from the environment as well as for the respiration purpose. Now if you look at woody stems, we all know what woody stems are. Now the type of stems, epidermis, the tissues that are found in the stems. Epidermis, the dermal tissue outside. Now it may contain a waxy layer that we find in the leaves. We call them cuticle, right? Or we may not have any waxy layer outside there. Or it can have a trichomes, like hairs of protection that release scents or oils, etc., that we just saw in the earlier picture, like this one. It may add some kind of oils, and you know, some cases this trichomes used as a rich source of some very sweet flavorings and provide some aromatic substances. If we talk about the stem vascular tissue, again it consists of xylem and phloem, but the arrangement of them are slightly different. The vascular bundle contains both, but the xylem, it again conducts water and provides a support through the stem. Phloem conducts food and also provides support. So this is a structure known as vascular cambium, because vascular cambium means this is formed with xylem tissue and phloem tissue. Specific bundles, we call it vascular cambium in case of dicot plants. As you know, it's a dicot plant, and this is the structure specifically kind of it's like a triangular structure like this, consisting of many different things like xylem, like phloem, and also some other stuff. You look at the vascular cambium, it occurs in woody stems only, and vascular cambium located in the middle of the vascular bundle between the xylem and phloem. So if you look at here, this portion is known as the vascular cambium, because you know phloem is present here at this region, xylems are present at this side. So between xylems and phloems there is a strip of cells present. It's giving a very rigid unit for only the woody type of plants, and we call them vascular cambium. And this is the example of vascular cambium in the middle, phloem cells at that side, xylem cells at this side. The example is given for the vascular tissues for trees, like bark at the outside. You know, we call tree means it's a huge plant, not very herbaceous plants, because you know in trees we have a bark outside completely surrounding every different tissues out there. We have a phloem in the middle, and then xylem at the here, and between xylem and phloem we have vascular cambium, and at the middle exactly we have wood that is mostly made up with ground tissues. Okay. Now if you talk about the griddling or cutting of tree, what you will find, if you cut a tree from outside, it will damage the phloem, it will damage the xylem, and ultimately it will eventually kill the tree. Now if you start cutting from outside, and if you chip slightly from outside and then leave it as it is, if not all the xylem and phloems are destroyed, the plant still will try to cope up with the situation, heal the situation, because you know if wound is introduced, then xylem cells become kind of alive, they'll start dividing. But if it's much difficult, I mean if we cut it in huge amount, there's nothing to be done by the plant, and plant will die. Now, there's an interesting fact about the vascular tissues because they form rings in the trees. Like they are called as annual rings. You know, we know of the annual rings. They are tremendously beautiful. It suggests that one ring equals to one specific year. Like as you begin with that, and each year's growth, there's addition of rings because the plant grows in two different ways. Plant grows in the tall or height. It also grows in the side. So it's a major growth and the lateral growth. Two types of growths are possible. So you see, these are the annual rings. The first year of growth, then the rainy season comes in, and actually not only you can see the growth, but also you can see how they're growing and what are the different environments they're encountering during their growth. For example, at the very first year, this is the very first year's growth. Then second, we have a rainy season. And how can you know about the rainy season? Because you see kind of a moistened wood in the middle. Then we have a very dark patch. It is going to tell us that there is some kind of burning of the tree. So it might be some sort of forest fire there. So you can actually you can see about all the years that a plant is growing every time, because the cells that newly added, and before, once the cells are added, the older skin stays there, so it's kind of dividing between the two new type of cells. For example, this one, it provides us the different timekeeping idea. It's called tree timekeeping, and actually people can do that in your house. It's a very good hobby and very interesting also. You can take some old tree, snap it out, and you can check some important things about that tree. But now it's not about you take a branch of the tree. It will not work. You need to take the mainstream of the tree's bark to finally see what's going on. But it's not good to rope a tree or kill a tree like that. So if you find any broken tree, you can get this idea of something, but it's not good to do it manually to get the idea. Because you see, this tree is very older, for 1489 when the tree is planted, and you see it's kind of very greenish. The first type of cell layers it's shown. Then we have Columbus lands in America 1492, 1620 Pilgrims and the Plymouth Mass, 1776 US independence, 1861 start of Civil War. You can see 1917 to 1945 tree survives 2 world wars through that time, and finally man lands on moon in 1969, and birth year of the idiot who cut down this tree at 1971. From that time the tree has been cut. So you see, this is the end point. I like this slide very much. I keep this. Now about the plant growth, because I've talked about xylem, we've talked about phloem, and we know the plant are growing in two different directions, a primary growth and a secondary growth, lateral growth and the apical growth. Both types of growths are important. And meristems, embryonic tissues that are called the stem cells. They are undifferentiated stem cells. They are totipotent. They can produce any cells of the plant body. They can divide and become any kind of cell. Now those meristematic cells will grow and divide. They are apical meristematic tissues are present who will divide and give the plant a growth and tall structure depending upon the genetics of the plants. Like two types of growth, like primary meristem, which are called as apical meristem, and secondary meristem called as a lateral meristem. Primary meristematic tissues, they make plant grow in the longer distance, like taller and taller, like the redwoods and pine forest trees. And they are present in the root tips and obviously in the shoot tips. While the secondary meristem make a plant grow in the wide, it's like the width of the stem, and they are more xylem, which becomes wood. They add more and more xylems together to make the wood much more strong. If we talk about, not going to talk about the apical tissue, because apical tissue and growth is kind of simple. Cell will divide and on and on and on and on to give a tall structure of the plants. But in the lateral growth is something to deal with that ear rings that we already saw. Like lateral growth, lateral meristems called as vascular cambium. We saw that vascular cambium earlier. What are vascular cambium? We saw. That vascular cambium has the functionality for the lateral growth, and they are present between the xylem and phloem. So what they do in this case, they're cylindrical structures throughout the place. Like as you see, these are the vascular cambium surrounding phloems at those sides and xylems at this side. Okay, so in this case, It forms secondary xylem on the inside and secondary phloem on the outside. So the idea would be something like that. So this is, say, the vascular cambium. Now, lineage of cells. So these are the phloem cells that are usually present. These are the xylem cells that are present. Now what they will do is that they will keep on adding. We take a color here. What is, let's take a different color. And it's going on that this vascular cambium will start adding secondary phloems there, and secondary xylems is there. They are secondary. These are primary. Okay. Just keep on adding secondary phloem and secondary xylem. Secondary phloem in the secondary phloem region, secondary xylem in the xylem side. They start adding these things. And slowly what happens, the secondary xylem on the inside slowly start converting into wood, and then they get pushed away, right? That's how they're kind of doing the stuff. The secondary xylems start becoming wood, okay? And only the most exterior xylem works to carry water. That is the idea. Rest of them becomes a part of the wood, and that's how it start engaging and becoming broad and broad, becoming wider in lateral events. Okay, this happens to the phloem. This happens to the xylem. Sorry, what happens to the phloem? Phloem becomes a part of the bark, while xylem becomes a part of the wood, because the outermost layer of the xylem only carries water, and rest of them becomes the wood. While phloem, from the outside, the vascular cambium, only newest xylem closest to the interior is alive and transports food. Not xylem, it should be phloem. The newest phloem that will produce is the closest to the interior, and they'll alive and transports food. Rest dies and slugs off and does not become a thick wood part, as you see. So this terminal portions of the xylem keep on taking water, and the phloems, the newer phloems only at the very terminal region, they also start carrying food, but rest of them are part of bark. So phloem becomes bark, xylems become a part of wood during the lateral growth. And if you want to know more about lateral growth and the mechanism behind lateral growth, you may watch my lateral growth video, and the link is provided in the description below. Now again, if you look at here, the cork cambium, so this is the idea provided. You know, this is cork cambium. It is a specific, very hard structure, forms outside the phloem and makes a cork cells which fill with suberin. Suberin is nothing but a waxy material that protect the trunk of and also the branches of the plant. And that is, you know, if rains and all the stuff, this is a waxy material for the prevention. So that's why you have a specialized cork cambium that is present. So this is the cork cambium at the end, and after that we have a living phloem. After that we have a vascular cambium, then we have a sapwood, and it contains the xylem part. Then we have the heart of the wood. Ground tissue is obviously the primary part of this hard tissue. Okay, so phloem becomes bark, and the cork cambium and cork is present with the phloem to give it a much more rigid and proper safe structure. Okay, now here if you look at the wooden structure from the wooden terminology, at the middle we have a very thick wood. We call it a heartwood. This is made with complete dead older xylems and filled with ground tissues. And we have a sapwood just surrounding the heart of the wood, or heartwood, and they are made with new xylem and they still carry water. That's why you call it sapwood, because they are kind of wet, they carry water, they're alive. Then we have a phloem which carries the food, right? And the older phloems become the part of the bark along with the cork cambium. So this is the structure of the wood, and how the wood and lateral development and lateral growth of the plants shape the woods and the stuff. So if we dissect the plants in different regions, you'll find those stuffs in different way. You see here, epidermis, cortex, then we have vascular cambium, and we have the xylems and phloems and place like that. And as you go to the bottom, it's much more layers, and it is going on like that. Okay. If we talk about the ground tissues, it will be a part of cortex and pith. It is also present in the stem. Ground tissues are very, very vital tissues, and it's a part of the pith or cortex. Cortex is outside surrounding, and the cortex for the stem is very thin most of the case, while the pith is very, very, very wide. As you see, the pith is very wide where the cortex is thinner. So you see, it's act to store foods, right? And sometimes it can act as a site for photosynthesis when it's green. Like this cortex, if I give it's a green cortex, it is filled with chlorophyll. The cells are alive. It can do a photosynthesis and produce sugar. While the other hand, if it's not functioning well, like the part of the pith, it stores food. Example is given here, potato, right? Normal potato is a part of the shoot, while in the other hand, the sweet potato is a part of the root. Now finally, the leaves, because we talked about root, we talked about shoot, now we need to talk about leaves. Leaves are also very, very important organ of a plant, because leaves, though it's very, very it's not very weighty, or it's not giving any structural support, but all the functional units, production of food is one of the most important thing that plant can do, and we don't. Now the function is photosynthesis, production of food for the whole plant body, all the cells of the plant body. And plant leaves have two different structures in the morphology. One is called as blade, and second one is known as the petiole. So if you look at the structure, this is the petiole, which holds the complete structure, that is called the blade. Blade is the surrounding region, the structure that holds all the cells for containing chlorophyll, and actually it increases the surface area so that gets more sunlight and can produce more food. Petiole is a stalk that connects the leaf to the stem to hold it properly towards the sun. If you look at the leaf anatomy, it is correlated with the photosynthesis, and its leaf anatomy is processed like a kitchen. It's a kitchen of a plant body where it produces its own food. It just requires carbon dioxide and water. It produces sugar and oxygen. That's very easy. And if you look at the schematic structure, they have an epidermal layer for both the sides, because it has two different leaflet units, though one side is front and one is back, but still it carries all this type of epidermal cells in both the sides. Now the photosynthesis may go on in both the sides. For most of it, some plants photosynthesis is only carried out at one side, while the other side photosynthesis is less and respiration works in high amounts. So we have a dermal. After that, we have a ground tissue layer. At the middle center, we have the vascular tissue, just like the structure of roots. So if you look at here, this is the center, and you see the tube-like structures, and this is nothing but, let me take a color here again. This is nothing but the tubing. This is the vascular layer, the vascular bundle that we see. Rest of the cells are present and arranged in stacks to hold the structures properly, while at the bottom part, the other side of the part which is not doing direct participations in the photosynthesis, filled with stomata cells that helps in opening and closing and taking up water from the environment. Now if you see lip epidermis, what you'll see, it is transparent. You see, this is the lip epidermis, one very thin layer of cells. Light can easily go through this, light can easily pass through this. In some cases it filled with kind of waxy materials, and we call it cuticle, which protects the plant cells and the leaf cells against drying, because plants survive in a very, very harsh conditions, like in huge temperatures through all the time and dryness. So it needs to survive with this cuticle layer. While the lower epidermis, it has a stomata with guard cells. Stomata is a small pores through which it uptakes water, and also it can take out carbon dioxide and leave oxygen outside. Okay, so this is how they involve in the stomata. It's very important thing to uptake the raw materials for photosynthesis. You see, stomata is the opening, and it is closed by two different types of cell. We call it a guard cells. Guard cells. If you look at here, the trichomes, it's also part of leaf epidermis. Trichomes are nothing but small extensions from the leaves also. Like, you know, we've seen extension in the root, extension in the shoot. Why not leaf? Leaf also carries this kind of extensions, but not all the leaves have that. Some of the leaves only have it. This is a beautiful illustration of staining. As you see, this is stomata and filled with guard cells surrounding the stomata to control the opening and closing of the stomata. Now, if we talk about the leaf vascular tissue, again, leaf vascular tissue is also made up with xylem and phloem, and it has a layer outside the xylem phloem combination. We call it a sheath. As you see, vascular sheath. Vascular sheath. So what we talk about, whatever veins we see inside, those veins are nothing but this vascular tissue for leaves. So it's very easy to understand about the vascular tissue by dissecting a leaf. And you see that in this leaf we have those vascular sheath, and inside we have two types of tissue, xylem and phloem. Veins are composed of xylem, phloem. Both of xylem carries water, phloem for the food transport. And these are the seeds. We call it bundle seed because they surround xylem and phloem to make it as a bundle of vascular cells. We call it vascular seeds or xylem seeds. They provide strength as well as support, and they organize xylem and phloem together. In some cases, having xylem and phloem together in close proximity is really, really important. That's why in all these cases of root structure and shoot structure, as well as in this leaf structure, we see that those things put together because they increase the stability and structural integrity of the plant cells, along with some kind of physiological function of carrying water and food also being helpful. If we look at the leaf mesophyll cells, mesophyll is, it's the middle of the leaf. We call it a mesophyll. Meso means the middle. So this is the rest of the part. It is made up with parenchymal tissues as well as ground cells. This is called ground tissues, but it's made up morely with parenchyma. Two types of parenchymal cells found in those leaf mesophyll middle tissue: palisade parenchyma and spongy parenchyma. Palisade parenchyma, they are long columns below the epidermis. So this is the epidermis. These are known as palisade parenchyma. As you see, long small rod-shaped cells that are found. And then we have a spongy parenchyma in the bottom, which are circular, and they have a lot of vacuoles. Extracellular space, not vacuoles, extracellular space in between. It's like a sponge-like structure with very small pores in the middle. While palisade parenchyma are organized very compact way. Palisade parenchyma helps in the photosynthesis, while spongy parenchyma, the spherical cells, we talked about it, and they have air space in the middle, and they are required for the gas exchange properly. Welcome back, friends. In this video we'll be talking about the plant water and sugar transport. So we've already talked about the morphology of plants. We've seen what are the cells that are making plants, the tissues that are the part of plant organs like root, shoot and leaves. Now in this we're going to know the physical function, the physiological functionalities of a plant cell. And in physiological functionalities there are two major functions to play. First is the production of food using photosynthesis, and second one is uptake of water from the soil. Both of them are very, very important from plant perspective. So if we begin with that, first thing here we are going to talk about is the water and sugar transport in plants. The first thing that I started discussion about this whole topic, I told you it's very, very stunning that how these large trees like redwood plants take up water from the soil and take it up to the 300-meter long, not 300 meter, 100 meter to 150 meter long trees, like 300 feet and over 300 feet long trees. That's a very interesting thing to know. Now here you will understand how they actually take water and nutrients through that place, as well as how they transfer the produced food, that is sugar, throughout the plant body. So water transport in plants, I can give you a simple analogy, just like you drink soda from a straw, right? What we do, we create a pressure, we take out something, we suck it, right? Suck means what we do, we take out the air. So if we take the air out, it creates pressure, so we call it a cohesive force. Force means molecules sticking to each other, and they create this linear chamber of molecules one after another as they are sticking to each other. Due to that force, they tend to attach to one another, and once if we bring one out, all of them will start coming out. And second force that is involving is adhesive force. Adhesive force means attaching of the molecules to the wall of xylem cells. Just like here, in this case, the cohesive force is between the soda molecules. In this case of plants, it's the water molecules. The cohesive force between different water molecules to hold them together, and the adhesive force between the water molecule and the wall of the xylem cells. Both of them works together to give us this idea. You see, waters involve with each other with the help of this cohesive force. This is the cohesive force that interacts water one with another. So if we bring one water, drag one water, rest of them will completely be dragged there. And adhesive force means they'll tend to interact to the wall of xylem. So here we have a theory to explain how plants uptake water. We call it transpiration cohesion theory for the transport of water through xylem. And why it's called transpiration cohesion? You know, cohesion is one of the biggest force that is involved to drag water, because the water molecules are attached with one another. And if I draw xylem like a pipe, the water molecules are attached with one another. So if I start dragging water from some force, if we apply some force to start taking up water and removing water from there, it will slowly start attaching to other water. So whole set of water molecules start to come out from the root region towards the top of the plant, because the water molecules are cohesively attached with each other. So this is the first thing that we need to do, that is to create a pressure, to create the pressure. And second thing is that due to that pressure, cohesive force will take action and the water molecules will move. So the first thing, we need evaporation to build that pressure. Evaporation of water through the leaves occurs through the stomata. You know, stomatal cells will open. Evaporation of water occurs and generates what we call is a sucking force that resembles to that of taking a soda. Just take the pipe in our mouth and suck the soda out. We are not actually sucking the soda out, we are sucking the air out. As a result, it creates a pressure of air. That pressure involves in the soda. It will push the soda molecules through the solution, through the pipe up to the top of your mouth. Same thing here, evaporation creates the force. Due to that force, I mean, as the water is leaving, it creates a sucking force, and that force creates the water molecules to move towards the forward direction, towards the top. And as water molecules are attached with one another, so a series of water molecules behaving like a column will move towards the top. Okay, that's the idea. And here we have two things here in the xylem and phloem. You know, in the plant leaves also we have xylem and phloem. Now the phloem, as you know, these are the cells that we easily take out water. As the water is evaporating, slowly they are going to take that. So if you look at the transport, how it looks like in the big picture, it's the chain of water molecules added one after another due to the cohesive force and moving from roots towards the leaves. Now the thing is, the energy comes from the sun, because sun applies the heat. Due to the heat, they need to go for the respiration. So if you ask me how this process is driven to the forward direction, I will call you the energy is provided by the sun to do that, because stomatal closing and opening is going on during the transpiration. And for that reason, if the water is lost a lot, in that case, to fill that out, water molecules will be going from the bottom towards the top due to that pressure. And cohesion will help them to be linearly arranged with each other, plus the adhesive force between the water and the wall of xylem will help them to properly guided towards the forward direction, towards the top. We call it a transpirational pull. It's the water vapor in the air space of the leaves that diffuse down its water potential gradient and exits the leaf via stomata. So the water potential gradient is getting down, and that causes the water to flow up. So what is water potential? We'll be seeing what is water potential in a moment, but this is the idea. We call it transpirational pull, because due to the transpiration, water is moved from the plant leaves. As a result, a force is built up and the water start coming from the soil towards the top of the plant. as you see in this case. It's coming from the xylem. Now transpiration produces negative pressure. We call it a tension in the leaves, right? And due to this negative pressure, the plant leaves try to get away from the pressure. How to minimize the pressure? It will start pulling water to the front. So these are the air-water interface. As you see, air-water interface, and as it's moving, water vapor lost in replaced. As you see, as it's moving, if you look at here, the complete structure is provided here. These are the water films, and this is the mesophyll cell, between the mesophyll cells, and waters are evaporating. Due to that negative force that is built up, a chain of water will start coming to the top, as you see it here. Cohesion and adhesion, we call this process ascent of sap. Sap is the water and nutrient combined water, so sap is coming, it's ascending, so you call it ascent of sap, and it is maintained due to two different forces: the cohesion forces between the water molecules and the adhesive forces between the water molecule and the xylem wall. So you see it here, water molecules are moving from the root, and as it's creating a pressure, the water molecule is driven towards that pressure to minimize that pressure, and they slowly start moving like a chain because they have a cohesive force and the adhesive force towards the wall, keeping it steady through the xylem tube towards the top. Okay, two types of force, adhesion and cohesion. And then finally they move to the top of the xylem, and they will be removed to the mesophyll cells, and they will migrate through the mesophyll cells finally, and through the stomata the water will go out again. So by this way, water keeps on moving from the plant, from the leaf using stomata, and that creates a pressure. To minimize that pressure, water flows back to the top towards the leaf. That's the idea. This is the overview of the whole process from the root here through the xylem of the shoot up to the level of the leaf. Okay, and if you look at here, there are some values given here. We call it psi. Psi is known as a water potential, and the water potential is varied for the different reasons from the different region of the plant tissue. For example, the water potential is very, very negative, very less. It goes very down at the leaf, and as a result of that, it creates the pressure. Then only it will force the water to move up. If the water potential is up there, then no way water is going to move. The idea about water potential is that water will flow from higher potential to the lower potential. See, the water potential at the root is minus 0.03, while the water potential at the plant leaf is minus 100. So it moves from the high to the low range. So see, what is water potential? Water potential is a measurement that combines the effect of solute concentration and pressure. The concentration of the solute and pressure is measured. Solute, in this case the water, and determines the direction of movement of the water. So water always tend to move from high water potential to low water potential. Okay, and in this case the high water potential is present in the root region, minus 0.03, while on the minus 100 in the leaves. Okay, so this is in the root, this is in the leaf. That is the reason water flows. Actually, we created this to understand the process very carefully. Like MPa is the unit to measure the water potential. We call it as a megapascal. Pascal is the idea of pressure. So it's nothing but how the water and solute applies the pressure. That is it. Because it depends on the pressure, guys. Because every time you think of plumbing and the system of how water flows between the pump, using pumps, and between the pipes, you always know if there is high pressure and there is low pressure, water is always going to flow from high pressure to low pressure. Now the thing is, normally if not respiration, if the plant is not going respiration, the water potential of the plant root and the water potential of the leaf is slightly different. The water potential at the root is always high because there is a lot of water concentration there, a lot of solute concentration there in the soil. So the water potential is always be high in the root section of the plant. But on the other hand, the leaf region of the plant does not have that much of water content. So obviously the leaf water potential will be low. The pressure of the water at the leaf will be low, that's for sure. But when the respiration is going on, the water is furthermore evaporated, creating very less concentration of solute at the leaf, which allows the water to come from high pressure region, which is root, towards the low pressure region, which is the leaf of the plant. That's how the water flows from root towards the plant. And we call it 0 MPa is for the pure water at the sea level at the room temperature. We call this water potential. Now, how solutes and pressure affect the water potential? Actually, water potential is the measurement of pressure created by the water. So obviously, if there is more solute, more pressure will be created, right? So we call it also as an osmotic potential, right? And there are different names like solute potential, which is known as suffix s of psi, and we know as pressure potential as suffix p for the psi. Pressure potential is the physical pressure on a solution that is applied towards the solution, while solute potential is proportional to the number of dissolved molecules that is present. So if you have high concentration of molecules dissolved, obviously solute potential is going to increase, and if solute potential increase, water potential also increase, because in this case water is acting there as a solute. And in this case, pressure potential is the potential that is applied on a solution, not applied by the solution, applied on the solution. That is pressure potential. And another thing we need to know is the turgor pressure. Turgor pressure is a measure, it's a pressure exerted by the plasma membrane against the cell wall, and that keeps the cell very much steady and very solid. That means, cell wall is present and cell membrane is inside. Now if there is not enough pressure to push the cell membrane towards the cell wall, then the cell, it looks like the cell will not stand straight, it will go down. And if you see, normally if you take some of the fruits and if you dehydrate that thing, if you take a vegetable, raw vegetable, or herbs from the market, and you put it in the dried condition, it will go down because the water will be evaporated, so there will be no water to create pressure to push the cell membrane towards the cell wall. As a result, it becomes flaccid. So what you need to do, if you put it into the water again, it will absorb some water, and again it becomes very solid because it becomes turbid. As the water will insert itself into the cell, it will create some pressure towards the cell membrane, towards the cell wall. So cell membrane will be pushed towards the cell wall, creates a high tension buildup, so they become turgor. So that's called turgor pressure, and those strong variety of cells and structures, we call them turgid. Okay, and we calculate this normal water potential, that is simple psi, equals to psi s plus psi p. Psi s is a solute potential, psi p is the pressure potential. So the water potential equals to solute potential plus the pressure potential. Now this is to explain you about this process in much more details. For example, there is a sucrose solution and a cell is placed inside that. When the sucrose solution is 0.4 molar sucrose solution with the pressure potential of 0 and solute potential of minus 0.9. Okay. Now let's say initial flaccid cell, if you take a cell which is living, healthy cell, initially the water potential was minus 0.07 Pascal, okay, megapascal there, minus 0.07. And now if we put it into the sucrose solution, 0.4 mole sucrose solution, in that case the water potential of that sucrose solution is less compared with that. So what will happen? The water potential is less outside, inside it's more, so water will flow from the cell to the solution. So the cell become, what will happen? The cell become dry, as it plasmolized. We call it a plasmolized cell. Why? Because in this case the cell will lose some water, so it becomes very plasmolized condition, so it's destroyed. Now it's not usually destroyed because the structure can be maintained in the future because they also have this cell wall. Remember, cell wall never does the shrinking and stuff because they are brick-like structure. The shrinking occurs of the cell membrane. Now, if I again take this cell from here and put it into the into a mixture where the solute potential or the water potential is higher, if you take it, put it in the solution whose water potential say 1. In that case, water will again flow from the solution inside the cell, pushing the cell membrane towards the cell wall, making the cell much more rigid again. Because of the cell wall, these plant cells are such different, I mean they are such a versatile to hold these situations. But if there are animal cells, there are no structural elements to give them any hard shape like cell wall. So in animal cells, if there is any plasmolysis, cell will burst open and everything will come out. But in case of plants, as there are cell walls covering the cell membrane, it will hold the structure. Okay. Now the another thing, if the same placid cell is placed in the solution, as you see here, the water potential is high, 0 Pascal here, 0 megapascal, and here inside is minus 0.75. So obviously water will flow from outside inside and make it more turgid. So obviously water will flow from high water potential to the low water potential. So that's about how water flows from the root, from the soil through the root up to the top to the leaves. Now we need to talk about how sugar is translocated. I mean, the sugar is produced in the leaves, and once the sugar is produced in the leaf, it should be distributed through different cells in there. So this is called sugar translocation or food translocation, whatever you say. It causes sugar made in the leaf mesophyll cells, because most of the time the mesophyll cells are the living cells. They carry all those necessary equipments for the photosynthesis like chlorophylls and stuff. So photosynthesis is going on in the mesophyll cells. So what happens now, once the sugar is produced inside the mesophyll cells, it will slowly diffuse to the phloem cells, in the vascular bundle of the phloem cells. And then what happens? Companion cell load dissolved sugar into the phloem STM using energy, and in this case they require active transport for this process to happen, because what happens, we are pushing something from high energies to from low energies to low concentration to high concentration, because you know these companion cells have low concentration there inside and high concentration there outside. So we need to go against the concentration gradient for sugar here. So we need to push it with some energy. ATP acts as an energy source to deliver it, and water moves into the cell with high sugar concentration due to osmosis. As osmotic water flow happens, so let's look at it in much more details in this picture. You see, this is the transportation pull. As a result, a pressure builds up and water slowly start to move through the xylem up to the top of the plant. And in this case, what happens, on the other hand, sugars are produced in these leaf cells, through which sugars are distributed towards those companion cells. But the thing is, companion cells, and through this these are the STM, the phloem transport cells, and those phloem cells we need to put them inside. So what we need to do, we are going against concentration gradient. So in that case, we need to apply some energy to put it back to the pressure, because the pressure is very high there. So we need to put a lot of pressure there. So red arrow means active transport we have to do. Though we have high sugar concentration there, but still due to the pressure difference, we need to add some force using ATP hydrolysis and put the sugars into the tubing made up with phloem. Now once we put our sugar into the phloem, and this is dissolved in water, and that water dissolved sugar can move and easily can flow down to the bottom, because now it will flow by basic rule from high concentration to low concentration. And once the food is prepared, the higher concentration will be towards the leaf, lower concentration will be towards the root. But all of the cells require food for their living. So once we put them here, during that process, water sometimes move towards the xylem. Why? Because of osmotic balance. Because you know, here there will be higher concentration of sugar, low concentration of water. So bring it back, water will flow from xylem to the phloem, and that is the water osmosis. It creates some osmotic pressure, so that thing keeps on going. So osmotic pressure is going to minimize the concentration so that the cells won't burst open, as well as the active transport is going on to push sugar to the nearby cells, and then finally down the concentration gradient sugar will flow to the later cells to the bottom of the plant. So see, these are the stages. An active transport is required, and this is how the active transport is that. We call it, in many plants, phloem loading requires active transport. As you see, there are proton pumps. For that, proton pumps are used, and they are very much effective because protons are pumped out. Once you add protons out using some ATP, because it's an active transport process, ATP hydrolysis provides the energy, protons is pumped out, and this proton is taken from the other side along with the sugar, and then we use the concentration gradient generated by proton to drag sucrose inside the cell. Otherwise, sucrose cannot directly insert inside the cell from those companion cells to the STM. Because STM is created by the phloem, and until and unless the sucrose is brought to the STM, the sucrose cannot be distributed, because this is the actual system to distribute food throughout the plant cells or throughout the plant body. And we have produced all those foods in the parenchymal cells, which is in direct contact with the companion cells. So from the parenchyma, sugars inserted into the companion cells. Companion cells use this process of proton gradient preparation, and then a co-transport machinery, a uniport machinery, using the proton gradient to take sucrose out to the STM. Once the sucrose is in the STM, then easily it can flow through the STM to rest of the cells in the plant body. Okay, so that is the overall process of how this thing works, how the food slowly distributed, and how water is taken. In both this case, it requires certain pressure and forces. You need to know what is diffusion, you need to know what is active transport, you need to know what is osmosis to understand the process. If you don't know about any of these terms, I would recommend you to pause the video and watch those videos in my channel first, then come back here and watch this. You'll be learning that very, very fast. So that's for it, guys. Thank you.